I'm pleased to introduce our next session, our next presenter. Rearing experience and genetic strain affect fear response in laying hen pullets. Anna Rentsch, University of Guelph. Please go ahead, Anna.
Thank you. I hope you can see this. All right.
Looks good.
Perfect. So aviary housings for laying hens are very complex, and the birds are usually housed in very, very large groups. Now, if we have a high level of fearfulness in these large groups, we do run the risk of injury and piling that can lead to smothering. Now, fearfulness by itself is not a bad thing. So fear is an adaptive anti-predator response, but it can lead to distress or injury when it's extreme or chronic. So in laying hens, we know of 4 different types of anti-predator responses: fighting, fleeing, freezing, and tonic immobility. I'm going to be focusing on fleeing and freezing today. So we know that there are some innate differences between genetic strains in fear responses and fear behavior in the laying hen. And we also know that environmental enrichment, as well as early life complexity, has been seen to reduce fearfulness. So we were asking, does the genetic strain affect fear responses to specific fear-inducing stimuli? And does the level of early life complexity affect fear responses? We were further interested in a possible gene by environment interaction. So do brown and white pullets respond differently to different rearing environments? To answer this question, we raised 4 flocks of 2 genetic strains, Lohmann brown lights and Lohmann selected leghorn lights. I'm just going to be calling them brown and white birds from now on. And we raised these 2 strains in 4 housing systems, but the 2 strains were always kept separately. So for the first 6 weeks of life, birds were housed either in conventional pullet cages or in 3 different types of rearing aviaries. And aviaries 1 to 3 increase in their environmental complexity, so the number of perches and platforms and space. However, they were all housed at commercial stocking density. At 6 weeks of age, the conventional birds, they stayed in conventional cages, while the aviary birds all gained access to litter and ramps and multiple tiers and perches. Again, the stocking density was at commercial level. And then when the pullets were 14 weeks of age, we did a fear test assessing fearfulness in a novel arena, novel object test combination. Here on the photo, you see the novel arena. The birds were tested in pairs, and video recorded behavior was coded from the videos and then analyzed in R&R Studio. Going forward, it's going to be important to realize that behavioral reactions to fearful events are only indicators of fear. There's no way for us to directly measure the subjective state of fear, but we do our best to assess fearfulness. So the novel arena portion of the test lasted for 10 minutes. The birds were placed into a circular novel arena. There were zones indicated on the floor and a feed dish in the middle. One of the 2 birds was always painted. That way we could code the behavior on an individual level. We scan sampled them every minute, and we were interested in 2 outcome variables. One being exploration, and we assessed that by counting the number of zone changes, with more zone changes being indicative of more exploration, which we would then assume means that they're less fearful. The second one would be alert behavior, so escape attempts, vocalization, alert, walking, standing, and running. After the 10 minutes, we would introduce a novel object. It would be lowered from the ceiling onto the food dish. And we were interested in the first immediate and involuntary reaction to the appearance of the novel object. We were also interested in a secondary response. For this, we scan sampled every 30 seconds for 5 minutes, and this response would be the adaptive and voluntary response to the presence of the novel object. Now, these responses can look like that. So for the first involuntary response, the birds could avoid the novel object. So they could flee by running away, walking, jumping, or flying away. Or they could freeze, so they could be stationary. However, they could also be indifferent, which could also look like stationary. And those 2 are really hard to disentangle. This is why we looked at that secondary response, where the birds could either remain frozen, so they would remain immobile, which we assume means that they're more fearful, or they could investigate and approach the novel object, which will be indicative of a lower fearfulness. So For the results, first I'm going to talk about the exploration in the novel arena. The strain differences are always going to be shown in green and the housing differences in blue. We have here on the y-axis the number of zone changes. So as you can see, there was no difference in exploration between brown and white pullets. However, birds raised in conventional cages explored less than birds raised in aviaries. There was no interaction. In terms of alert behavior, here on the y-axis, the count of alert behavior, we saw that brown pullets showed numerically more alert behavior than white pullets, with a statistical difference, and birds from conventional cages too showed more alert behavior than birds raised in conventional cages. There was again no interactive effect. For our results in the novel object test, so here we have the avoidance as an odds ratio and the approach, the secondary response, as a hazard ratio. The white birds will always be compared to the browns and the aviary birds to the conventionals. So what we see here is that white birds were 2.6 times more likely to avoid the novel object as the initial response, but there was no difference in approach. Aviary birds from aviary styles 1 and 2 were more likely to avoid the novel object initially than conventional cage birds, and birds from aviary style 3 remained immobile to the same amount as conventional birds.
Yeah.
Now, birds from aviaries were more likely to approach the novel object as a secondary response, and there was again no interactive effect. So what does this mean for birds in terms of life in an aviary? So we saw that brown birds were more alert in the novel arena, but explored it equally than whites. And we also saw that browns are freezers and whites are fleers. This has been shown previously by work from our lab. Now, this does not necessarily mean that whites were more afraid than browns. They could be equally afraid, just have a different reaction. However, them being fleers and living in an aviary increases their risk of injury by collision with metal structures, for example. So we could say that brown pullets might be more alert in a novel situation, but their response to a novel stimuli could be more adaptive to life in an aviary by decreasing their risk of injury. In terms of housing differences, we saw that conventional cage pullets were more alert in the novel arena, they explored it less, and they were freezers and remained frozen as a reaction to the novel object. This makes us think that they were more fearful in both situations. Birds raised in an aviary 1 and 2, so the low and intermediate complex aviaries, were fleers, and aviary style 3 birds were freezers, or maybe they were indifferent. It's really hard to tell. However, all aviary birds recovered and approached the novel object. Again, being fleers doesn't make you necessarily more afraid, but it increases the risk of injury in an aviary. So from this, we could potentially say that birds raised in an aviary style 3 might show an adaptive response to life in an aviary. They don't respond through fleeing, and they don't seem to have a high level of fearfulness towards these two situations. While birds raised in conventional cages might actually have an adaptive response to life in a cage. So they're freezers, so they're not as likely to get hurt. And them being more afraid towards novel stimuli, it's not that big of a deal, seeing that they're probably not going to encounter any novel objects. So from this, we could take home that genetic strain did affect fear responses to fear-inducing stimuli by brown and white birds having different responses, even though it's not clear if one or the other were more afraid. And the level of early life complexity did affect fear responses, with birds from conventional cages being or appearing more fearful in both situations, and birds raised in a most complex aviary having a different fear response to birds raised in low or intermediate complexity. We did not see any gene by environment interactions, so brown and white pullets did not respond differently to the different rearing environments. And with this, I would like to thank you all for your attention. I would like to thank my advisor, Tina Wadowski, the staff at the research station, and all the people and mostly students that helped me collect my data. I would also like to thank all the funding agencies that made this project possible, and please let me know if you have any questions.
Thank you, Anna. We do have one question to start us off. That gives the rest of you time to queue up the rest of yours, so get typing, folks. So, did you measure any other parameters to correlate, for example, freezing behavior with fear?
I did not. The fact that freezing is a fear response has been well documented in laying hens, so we can be pretty sure that freezing in itself would be a fear response. However, I did point it out, and I would like to point it out again, whether these birds here were frozen in fear or just stationary because they were indifferent was not visible based on their first reaction. So we could not discern that. However, based on the secondary response, it was pretty obvious then that birds from conventional cages, for example, were probably frozen in fear, seeing as they remained stationary for the whole 5 minutes of the test. But no, we didn't like collect any court measures or anything like that, if that was the question.
Okay. Two more questions. I was just wondering how many birds were involved in this research.
So in this specific test, I think, what was it, about 160 pairs each? Let me go back to this. Yeah, 170 pairs each, 172. So times 2 by strain, times 2 because they're pairs. In the whole project, there was a lot more birds. So we raised those 4 flocks, and each flock had 3,000 birds. We did a bunch of other tests with them, however, we weren't just interested in fearfulness. We looked at locomotion differences. We looked at general behavior differences, cognitive differences between the different housing, etc.
Perfect. We have another question. Whites appear to be fitter than browns and more physically capable. Could the physical capability or willingness influence the freeze versus flee?
Well, I suppose capable, I'm not sure. It's a can't or won't kind of question. So we see that white birds appear more active. They locomote more in their environments. They're often more active in behavior tests. I'm not sure if they're more capable or if brown birds just are less motivated. So it has been shown previously in the literature, and it's also what I found in most of my tests, that brown birds are less motivated by food. They are less motivated by social contact. They're overall a bit less active, which might be due to their lower motivation by cues for foraging or social interactions. It doesn't necessarily mean that they're less capable. It's just they might not choose to show what they can do. So this is really, really hard to disentangle those two things. And yes, whites being a bit more active might show here in that initial fleeing response that they perform. I think based on our aviary responses or housing responses, there is a hope that the early life environment and early life experience could maybe change their initial reaction of the white birds that tend to flee to maybe not flee as extremely. So that could be good for them, or good in terms of injuries like killbone damage or things like that. But yes, they behave differently in almost everything, so it's no surprise that they were different in their fear responses. I think when it comes to housing systems, that might be something that we should maybe consider. Are the same rules applicable to all strains?
Thank you. I am waiting for any other questions to come up. Very quickly, as we do have a few seconds here, what are the applications to industry of your current findings?
So in Canada specifically, egg farmers have chosen to move towards open systems, so aviary systems or enriched cages, and go away from conventional cages. Now, as we're housing laying hens in these complex housing systems, the question arises, how do we rear the pullets? And what I'm hoping to do is, with all the different tests that I did, sort of build a body of knowledge on how the pullets develop in these different systems, also based on strain, And this could then be used as information when farmers want to make decisions on maybe what strain to rear or what housing system to rear their birds in. It's not necessarily to point towards a specific system and say, oh, this is the best one, but more to show how do the birds develop depending on genetic strain in these different systems. What do we see?
Thank you. Very comprehensive. So we are going to leave it there. Thank you very much for your presentation and for handling the questions.
Thank you so much. Bye.